today we are going to talk about mitosis. Yesterday we talked about the cell cycle, and I figured doing it in a different way than just like writing and drawing would give us a little bit of spice for our Wednesday. So we are going to make these foldables with these flaps with all the information on them. So first we're going to do some folding. So big old piece of paper, fold it in half, hamburger style, sorry, yeah, hamburger style, to where you literally just have like a normal sized piece of paper. So if you're doing this, you could do this with 2 normal sized pieces of paper if you're just like, you just have it in 2 parts instead of 1. Okay. When you are finished with that, you're going to fold this into thirds. The way I found to do this is to kind of roll it, get it where you want it to be, and then just kind of like chest compression it down. It's not going to be perfect, but it'll be pretty darn close. When you open it up, you should have 6 different columns on your paper. Okay. So when you fold it into thirds this way. That way, yep. Okay. Then, when you got it all unfolded, and you have your 6 segments, you can take the bottom of the page and fold it up, but not all the way up, so you still have room for the titles, and then you have a flap. Huh? Yeah. Yeah, no problem. Ta-da. All right. Y'all want rulers? What I did was we have to cut on the lines and like separate them out. So I used a ruler, but you can just freehand it if you want. So okay, so I just went against all of the folds that you have into the 6 sections. Go ahead and divide those out. I did it while it is folded up first, just so you can clearly see the 6 columns. And then I did the same thing on the inside, just so I can differentiate. My scissors over here. Yep, and then y'all are already on it. I went through and cut along the lines that were on like the just up till that crease for on the shorter half. We will be drawing the phases of mitosis. So if you are a colored pen person and you don't have them on you, you can use the colored pencils that are on my table over there. All right. So when we're done with this, you can glue it in your notes if you'd like by folding it in half. Whoop. Where am I? Just folding one half, gluing it in here, and that way it can open up that way, or probably that way. Just so you have it and you don't lose it. And I like flaps and stuff in notebooks. So now we have a pretty fun little foldable thing. All right, here we go. So you have six segments, 6 titles across the top. Well, you are going to name them. The first one is going to be interphase, specifically G2 of interphase. We are skipping G1 and we are skipping S. So this is G2 of interphase. So where we are starting is after our cell has grown and after our DNA has replicated. The second phase we are going to touch is prophase, then pro-metaphase, which is a new one. A lot of so if you have only ever taken like an intro bio before, usually it's interphase, prophase, metaphase, anaphase, telophase, cytokinesis. We have added a new in-between step, and that is pro-metaphase. So we've got prophase, pro-metaphase, metaphase, anaphase, and the last one is going to be telophase and cytokinesis. G2 of interphase, prophase, pro-metaphase, metaphase, anaphase, and then the last one is telophase and cytokinesis. Now, there are a couple ways we can do this. We can go through, we can draw all the pictures first, and then go back through and write on the inside of the flaps, or we can take it one flap at a time. Do any of you have a preference? Would anyone like to make the decision? One flap at a time, that works for me. So, y'all, okay. All right. If we don't get through them all today, it'll be fine. We can finish it up tomorrow. Nothing too crazy. So, starting off strong with G2 of interphase. Go ahead and on the outside of the flap, we're going to draw just a big old circle. Ooh, that's fun. Yep, outside. Circle. We're going to draw a little nucleus. Ah. Okay. Okay. So we've got the outside of the cell. This inner circle right here is the nuclear envelope. Inside the nuclear envelope, we have chromatin. Remember from yesterday, chromatin is all of the DNA and all of the associated proteins that a chromosome is built out of. A lot of C words. The inside of all that, there is a darker region. That is the nucleolus. Okay. Then we have centrosomes. So each pair of these little squiggles is a centrosome because each individual little each one of these little tic tac looking things is a centriole. So two centrioles make a centrosome. A centrosome has that little squiggly around it. They are duplicating at this point. And then we have our nucleus with our nucleolus and our condensed chromatin. And that's our picture for interphase. Nothing too crazy. So on the inside, I've got it written like bullet points. Nothing too intense. Just what's happening during this phase. Be cool if I. Okay. At this point, our nuclear envelope encloses the nucleus. Our nucleus has one or more nucleoli or nucleolus. So the plural of nucleolus is nucleoli, which can kind of be a tongue twister. Two centrosomes have formed by duplication of a single centrosome. So if you remember, G2 phase is our prep phase, where we are making all of our extra organelles for cell division. A centrosome is considered an organelle, so it is also duplicated in this phase. The definition of a centrosome, just so you have it, is a region in animal cells that organize microtubules. Region in animal cells that organize microtubules. Plant cells don't have centrosomes. Plant cells have spindle fibers. Plant cells have microtubules, but they don't have this organization center. Each centrosome has 2 centrioles. Each centrosome has 2 centrioles. And our chromosomes are duplicated but not condensed. That's where we are with G2 of interphase. All right. Doing okay? Okay. Moving on to prophase. Just circle on the outside again. Okay. Just like last time, I'm not going to get the circle tool out, but we still have a nuclear envelope. It's still intact. I'm going to make that a little bit bigger just for me. In our nucleus, our chromosomes are starting to condense, but they're going to be a little wigglier, a little longer, a little spindlier than normal. So I'm going to draw 2, or I'm going to go with 4, just to have our multiple chromosomes. So we've got 4 wiggly chromosomes in our nucleus. Where these 2 chromosomes meet, if you remember, is called the centromere. These little lines are going to eventually become the mitotic spindle. So we are going to call this the early mitotic spindle. And this little star-looking thing around the centrosome is called an aster. Aster is, I think it's Greek, but I could be lying. Another word for star because it looks like a star. So that little like wiggly line thing around our centrosome is called an aster. I guess I could have made the chromosomes the same color as the chromatin from the first one. Whoops. Okay. That is our prophase drawing. I believe. Yeah. Prophase and metaphase are a little bit longer, but I swear they get shorter as we go on. All this prep. We got lots of stuff to do. So in prophase, we qualify. Okay. In prophase, our chromatin condenses into discrete chromosomes. Chromatin condenses into discrete chromosomes. Our nucleoli disappear. Each chromosome has 2 sister chromatids. Each chromosome has 2 sister chromatids. Our mitotic spindle begins to form. And it begins to form out of the centrosomes and microtubules that extend from the centrosomes. So our mitotic spindle begins to form out of the centrosomes and the microtubules that extend from those centrosomes. Just like we labeled on our picture, those radial arrays of microtubules, those little stars, are called asters. Radial arrays of microtubules that extend from centrosomes called asters. And finally, what's happening here is that the centrosomes move away from one another, and they are propelled by the lengthening of those microtubules. So as those microtubules that attach to them extend, as they get bigger, the centrosomes will move. Centrosomes move away from one another. Propelled by extension of microtubules. The next phase is pro-metaphase. And they kind of added this phase between them as we kind of get a bigger understanding of what mitosis is. They have not necessarily discovered, but kind of delineated that at some point in time between prophase and between metaphase, there's an in-between state that we can call its own phase, which is pro-metaphase. prometaphase. Get my circle out. I'm really glad I figured out how to do this. Okay, really big one, but that's all right. During prometaphase, okay, our asters are pretty darn close to at either end of the cell. Chromosomes I had randomly changed to another color. That's all right. Our chromosomes have started to move, but not quite move. Our nuclear envelope is all broken up, but not quite gone. And our spindle fibers have started to connect. All right. So that's what we're looking at. Still got it. All right. So we've got a little bit of a longer one, but we can do it. All right. So during prometaphase, our nuclear envelope has fragmented. Nuclear envelope fragments. That allows our microtubules to invade our nuclear area. Usually our chromosomes are protected from the outside of the cell, but as that nuclear envelope deteriorates, our microtubules can get in. Our chromosomes are a lot more condensed. That's why I drew them less wiggly. And then we have this new thing called a kinetochore. Kinetochores are specialized proteins and they form at the centromere of each chromosome, 2 per chromosome. And at the end of some microtubules. So we've got these kinetochores that are special proteins that help connect things, if you will, or help kinetic movement. They are at the center of each chromosome, so at the centromere. There's 2 per chromosome, and at the end of some microtubules. When a microtubule has a kinetochore, it is called a kinetochore microtubule. Yes, so the microtubules attached to kinetochores jerk chromosomes back and forth. So some microtubules connect to kinetochores of chromosomes and jerk chromosomes back and forth. So a lot of the graphic depictions of mitosis show like smooth movements, but it's a lot of just like pulling them one way, pushing them another, just kind of randomly tugging them until they get where they need to go. But there are some microtubules that aren't attached to kinetochores, and they interact with microtubules from the opposite end of the cell and work to lengthen the cell. That's how the cell gets a lot wider. So non-kinetochore microtubules non-kinetochore microtubules interact with microtubules from the other end of the cell. This lengthens the cell. Boots are squeaky today. All right. Ready for metaphase? All right, we're halfway there. Metaphase. Okay. In metaphase, we have our centrosomes at either end of the cell. We have our chromosomes lined up in the middle. I don't know why it's so hard for me to do that. And we've got our microtubules connected to the center of each chromosome by kinetochores. But we can't forget the ones that interact with one another and the ones that are just here to help out. If we were to draw a line directly down the center of the cell, equidistant from either pole, that is called the metaphase plate. Metaphase and anaphase, I think, are like the most familiar looking phases from regular bio. All right. So that is our metaphase picture. During this phase, our centrosomes are at either pole of the cell. They've officially made it there. Centrosomes are at either pole of the cell. Our chromosomes have arrived at the metaphase plate. And the metaphase plate, just to give you a definition, is a plane equidistant from both the poles. Usually when we see mitosis drawn out, it is in 2 dimensions, so it's kind of hard to see this as a sphere. But remember, this is all happening in a 3D space, so the metaphase plate would literally be like look like an actual plate, just in the middle of like a sphere. The centromeres of chromosomes are at the plate. Centromeres of chromosomes are at the plate. Okay. So, bringing the kinetochores back into this. So, for every single one of the chromosomes, so in our pictures we've got 4, in our bodies we've got 46, right? For every single one of those chromosomes, the kinetochores of the sister chromatids, so there's 2 kinetochores at each centromere, the kinetochores on each sister chromatid are attached to the kinetochores at the end of microtubules coming from opposite ends of the cell. So the microtubules with kinetochores attached to the kinetochores on the centromeres of the chromatids. A lot of C words. So, for each chromosome, oh no, I ran out of space? Goodness gracious. For each chromosome, kinetochores of sister chromatids attach to kinetochores of microtubules from either end of the cell. All right. I think we have time to draw anaphase. Actually, you have a lot of stuff to put away. Yep. We'll do anaphase, telophase, and cytokinesis tomorrow. Before picking up with what are we doing? Goodness gracious. Tomorrow is supposed to be and it's a shorter lesson too, so we shouldn't be too bad on time. Cytokinesis, binary fission, and then we've got another evolution connection. So nothing too crazy. We will finish up anaphase, telophase, and cytokinesis. Go a little bit deeper into cytokinesis. Talk about how the division process works in prokaryotes with binary fission, and then finish up with some evolution connections. Then Friday, we talk about cell cycle regulation and control. Monday, we will talk about what happens when cells don't listen to those controls.